In an unparalleled tour de force of biomedical ingenuity, researchers from the Guangzhou Institutes of Biomedicine and Health have monumentally transcended existing frontiers in organ transplantation by successfully engendering humanized kidneys within the biological milieu of porcine embryos. Disseminated in the venerated scientific journal Cell Stem Cell, this seminal work emerges as an avant-garde exemplar in the quest for efficacious and novel therapeutic modalities for renal pathologies. The apotheosis of chimeric biogenesis. This prodigious advancement epitomizes a quantum leap in chimeric biogenesis. While antecedent research had been circumscribed to the cultivation of comparatively elementary human tissues in non-human milieus, this equipoise of scientific and clinical sagacity has accomplished the cultivation of a meticulously structured, humanized renal organ within an interspecies framework. Liang Shui Lai, a senior luminary in the cadre of authors, elucidates, our sui generis approach circumvents the extant vicissitudes intrinsic to the orchestration of human cellular integration into non-homologous biological matrices, thus catalyzing the prospective vicissitude of in vivo human organogenesis within porcine hosts. The intricacies of methodological alchemy, a triad of ingenuity. The consummate actualization of this enterprise was predicated on a triptych of methodological paradigms. Ontological vacuums. Employing CRISPR Cas9 technology, the research consortium engendered a cellular lacuna within the porcine embryo. This ingeniously constituted a congenial ontological milieu within which human cellular entities could proliferate devoid of interspecific competition. Genomic alchemy of pluripotent stem cells. Human pluripotent stem cells underwent an elaborate regime of genomic modifications, thereby transcending their inherent limitations in apoptotic susceptibility and optimizing their predisposition towards embryonic assimilation. Harmonization of cellular ecologies. Subsequent to cellular engineering, the chimeric embryos were incubated within a bespoke culture medium. This medium was calibrated to satiate the incongruous metabolic requisites of the disparate cellular taxa involved. Empirical illumination. The substantive realization of theoretical postulations. Out of 1,820 embryos meticulously transferred into an ensemble of 13 surrogate sows, a quintet were extracted for thorough ontological scrutiny. These revealed kidneys in the Mesonephric stage, extraordinarily composed of an amalgam of 50 to 60% human cellular constituents. The nephric architecture manifested fully formed nephronic tubules and nascent uretic buds, incipient conduits to the future urinary bladder. Zhen Dai, another senior author, avers, by sculpting a bespoke cellular niche within the porcine ontological framework, we facilitated a symbiotic cohabitation, thereby maximizing the propensity for human cellular colonization. Significantly, ethical ramifications were circumscribed by confining the interspecific cellular contributions solely to the kidneys, forestalling any deleterious issues surrounding human cell integration within neural or germline tissues. Perspective horizons and complexity. The next strategic ambit includes prolonging gestational durations for enhanced nephric development and the application of this technological syntax to other vital organs. Nonetheless, the intricacies for real-world clinical deployment are labyrinthine. To craft an organ replete with multiple human cellular lineages necessitates a quantum leap in our ability to engineer the porcine host in a stratified manner, states Miguel A. Esteban, another senior contributor. Meanwhile, the current exegesis of this paradigm serves as an invaluable heuristic modality for studying human organogenesis and pathophysiological etiologies. This is an irreplaceable crucible for delving into human developmental biology, concludes Esteban. With this landmark publication, the Guangzhou Institutes of Biomedicine and Health have indelibly inscribed their name in the annals of medical science. This investigational feat not only augments our profundity of understanding in developmental biology but also heralds the dawn of an era where organ insufficiency could potentially be obviated.